Hello everyone. My research project is disrupting the sarcomeric mitochondrial creatine kinase into different oligomeric states. Creatine kinase is an important enzyme that catalyzes the reversible conversion of creatine and ATP to ADP and phosphocreatine. What this does is that it generates a large pool of phosphocreatine in the cell, which then allows the cell to readily generate ATP at times of high energy demands. There are a total of 5 different isozymes of creatine kinase, 3 of which are formed as dimers and are called the muscle type, the heart type, and the brain type, and these are all localized in the cytosol. More interestingly, however, the other 2 isozymes that are formed of creatine kinase are formed as octamers, the first one being the sarcomeric mitochondrial creatine kinase that is strictly found in striated muscle tissue, and the second one being the ubiquitous mitochondrial creatine kinase that is mainly found in the brain and in many energetically demanding tissues. The octameric state of creatine kinase was found to be crucial for mitochondrial binding and proper functioning of its catalytic properties. Thus, it is important to be able to characterize the different oligomeric states of this enzyme to further understand its relation with the cell and its possible implications in disease. For this research project, we will be focusing on the human sarcomeric mitochondrial creatine kinase. From a characterizing point of view, there are 3 oligomers of interest: the octamer, which exists naturally, the dimer, which is found to exist in low amounts compared to the octamer, and the monomer as well, which can only arise in harsh conditions. To be able to obtain the monomer, our research strategy was to identify potential amino acid residues in the protein crystal structure that may be important in the formation of dimers. These residues were located at the dimer interface, as indicated by the red dashed line. Important amino acid residues interacting with the adjacent protein subunits are expected to have a distance of less than 2.5 angstroms, which is the approximate distance of a hydrogen bond. By deleting these residues, we would expect to obtain our target enzyme in the monomeric form, because these residues help stabilize the dimer interface. Multiple sites of interaction were found on the dimer interface, from side chain to side chain interactions shown on the right, to side chain to backbone interactions shown in the bottom left. Many residues were thought to be important in dimerization and were then targeted for further analysis. The collection of residues that were found on the dimer interface that were thought to be important for dimerization were also found on the other side of the dimer interface. So what this means is that there's a two-point interaction that further stabilizes the dimer. The octamerization of creatine kinase occurs between the combination of 4 different dimers. Thus, disrupting the octamer interface would yield the dimeric creatine kinase. Much like the dimer interface, a collection of amino acids were found to exist in the octamer interface that were thought to stabilize the octamer interface. These amino acids were targeted as well for further analysis. Again, the collection of amino acids identified was found to exist on the opposite side of the octamer interface, which then ultimately stabilizes the octameric state. Additionally, since 4 dimers combine to form the octamer, each dimer will have a total of 2 adjacent dimers interacting to stabilize the octameric state, where each adjacent dimer are placed on opposite sides of a dimer. Thus, the same collection of amino acids were also found to exist on the other side of a dimer in another octamer interface. Ultimately, these symmetrical interactions help stabilize the octamer greatly. The next step is to mutate the amino acids found to interact in the dimer interface and the octamer interface to alanine, which is a small nonpolar amino acid that is incapable of forming strong intermolecular interactions. These mutants were purified and checked for its oligomeric state in solution to confirm if these identified residues were indeed important for overall oligomerization. 8 mutants were introduced that were aimed to produce monomers, and the other 4 mutants introduced were aimed to produce dimers. Double mutants were also included to ensure the complete disruption of the interface. All 12 mutants, along with the wild type, were expressed in E. coli and purified. Extracted proteins were tested for purity on STS page, an electrophoresis method that separates proteins based on size in denaturing conditions. All mutants and wild type gave similar bands and were in agreement with the molecular weight of creatine kinase in literature, which is about 46.5 kilodalton. Next, each mutant and wild type were separated in size exclusion chromatography, otherwise known as gel filtration, which separates mixtures based on size. Large and bulky molecules tend to elute first before smaller molecules. As shown below, the wild type, which is an octamer, eluted faster than the mutants which were designed to form monomers. Thus, this tells us that the mutants generated were able to disrupt the dimer interface. Some mutants completely disrupted the octamer into monomers, while some mutants yielded two peaks in the gel filtration profile, which tells us that there was a partial disruption of octamer into monomers. Meanwhile, one mutant was unsuccessful out of the 8 generated. We can also compare the stability of the different oligomeric states by finding the melting point of each protein, which is the temperature at which the number of folded proteins in solution is equal to the number of unfolded proteins. The higher their melting point, the more stable the protein is. Differential scanning fluorimetry, or DSF for short, is a technique used to measure the melting point. The technique uses a hydrophobic protein dye called cyperorange to monitor protein unfolding as we ramp up the temperature. Upon binding, the dye is unquenched and fluoresces. The resulting light can be detected and quantified to calculate the melting point and to monitor the transition of proteins from the folded state to the unfolded state. The mutant monomers were found to have a lower stability than the wild type. As shown in the transition curves below, the transitions of the monomer mutants were much earlier compared to the wild type. The mutant monomers had a lower melting point of about 4 to 5 degrees Celsius compared to the wild type. Moving on to the mutants aimed to produce dimers, only one mutant generated a dimer. That mutant yielded a peak on the gel filtration chromatogram that had a retention time in between the octamer and the monomer. This leads us to believe that this mutant was successfully able to disrupt the octamer interface to produce dimers. The other 3 mutants produced peaks similar to that of the wild type. Comparing the thermodynamic stability of the dimer to the octameric wild type, the dimer mutant had a slight decrease in stability by approximately 1 and 1.5 degrees Celsius, much less compared to the thermodynamic stability decrease found in the monomers. Finally, finding the melting points of the mutants with no change in the oligomeric state had no change in the thermodynamic stability either. It would also be interesting to compare the catalytic properties of each oligomer. However, monitoring the reaction spectroscopically is a challenge since all products and substrates have overlapping absorbance spectra. So, we designed a coupled continuous assay that combines our enzymatic reactions to other enzymatic reactions, such that only a single type of substrate in the assay can be monitored. We coupled our enzymatic reaction to pyruvate kinase, which takes the phosphocreatine and ADP to produce ATP and pyruvate. Next, another enzymatic reaction is coupled such that lactate dehydrogenase will consume pyruvate and NADH to produce lactic acid and NAD+. NADH is a substrate that has an absorbance wavelength of 340 nanometers. No other substrate in the assay has the same absorbance wavelength. Thus, monitoring the change in concentration of NADH will help us monitor the rate of the creatine kinase reaction. By running the assay, we titrated the enzymes from 0.5 micromolar to 3 micromolar and measured the rates accordingly. The rates were then modeled according to the Michaelis-Menten equation, which then yields a hyperbolic fitting. It was found that the octamer was found to catalyze the reaction faster than the dimer and the monomer. In conclusion, we were able to successfully disrupt the octameric proteins to dimers and monomers. Additionally, we were also able to see a clear shift in thermodynamic and kinetic properties between the octamers, dimers, and monomers. However, there is still plenty of room for further analysis. For instance, we hope to perform differential scanning calorimetry to employ a more absolute technique in measuring the melting point and to determine the enthalpy change of unfolding. We can also use circular dichroism spectroscopy to measure the rate of unfolding. Lastly, more elaborate initial velocity studies can be performed to compare many kinetic properties between oligomeric states, like the Michaelis constant to measure substrate affinity, or even measure the catalytic efficiencies of each oligomeric state. Thank you for taking the time to listen to my video on my research project on creatine kinase, and I thank you for giving me this research opportunity.